Good day sa inyong mga beloved students of Cebu Normal University, and welcome na po sa ato ang another class session sa ato ang subject nga biotechnology. Okay, so this is just a continuation sa ato ang discussion previously. So last nato nga video sa atong biotech nga subject is we talk about the historical and the philosophical foundation of biotechnology. So we learned that biotechnology has already been practiced during the ancient times. So biotechnology is not relatively a new science, only that there are technologies that make biotechnology nga na-modernize siya, and makita na to that there are several impacts, positive impacts nga ato ang na-benefit from the development of biotechnology. Okay, so this morning we are going to discuss the second topic. Okay, so mo na nga kato atong gi-mention previously nga when you say biotechnology is that there is really a heavy portion where molecular genetics is actually being discussed. Okay, so kana siya. So that is why maybe some of you, this is already a review sa inyong previous nga mga subjects. This is actually the molecular biology, the central dogma of molecular biology. Okay, so in the next 40 or 50 minutes, we are going to discuss the said topic. Okay, so let me just shift the screen. Ayan. Okay, so that's topic number 2. So the central dogma of life. Okay, so this will be the subtopics that we will discuss. The DNA structure, we will revisit the chromosomal structure of DNA. Then we also go to the molecular structure, and after that, the second subtopic will be the central dogma, and we discuss replication, transcription, and of course translation. Okay, so when we talk about the DNA structure, take note that DNA is actually known as the hereditary material of every, take note that DNA is the hereditary material of every known organism. So far sa biology, okay, there is not a single organism nga wala gi-operate sa DNA, because we know sa genetics that DNA is the blueprint of every organism here on earth. So different species, though different ang iyahang sequences, but relatively the structure, the foundation is the same. Okay, wherever you go, bisan dagko nga mga animals or mga microscopic animals or microscopic organisms, they are actually made up or being operated by the DNA. So take note that it is the blueprint of identity. So kung unsa ang naa sa imong DNA is mao na ang mugawas. Di ba, kung unsa ang naa sa imong genotype is mao ang mugawas sa imong phenotype. Okay, it's just like the blueprint of kanang gitawag natong mga buildings, di ba? We talk about the CCLX. For sure, CCLX have the blueprint. We talk about those skyscrapers. Naa na siya mga gitawag nato nga mga blueprint. So take note also that DNA is the language or code of cells that oversees the operations of every cell. So you can also say that DNA is mo na siyang kanabitawang manual. Okay? Manual of operation, 'di ba? So even sa ato ang school, mga companies na agyo na siya yung mga manuals. Okay, din na nimo makita unsaon pag-operate, what are the mechanics or the mechanisms on how every department operates, 'di ba? Say for example sa kanang student code, 'di ba? Kanang student code, I'm sure that you were given copies of the student code. So dito nito makita kung unsa ang mga degree programs, unsa ang pag-compute sa grades, so on and so forth. Okay? So others call it the blueprint of our organism. So that's what I mentioned a while ago. Okay? So most DNA are located inside the nucleus, particularly sa eukaryotes, di ba? So especially sa eukaryotes and sa cytoplasm if we refer to the prokaryotes. But regardless whether an organism is a prokaryote or a eukaryote, still DNA, the foundation of DNA, the concept of DNA is still the same. Okay? So some are found, okay, wala din makita. Okay, so some are found inside the mitochondrial DNA, inside the mitochondria. So, in the eukaryotes, take note that the mitochondria is somewhat special because it contains the genetic code na siya gitawag mitochondrial DNA, and it's actually an interesting field of study po, especially when we study genetics and heredity. Okay, so that is why there are some who speculated that mitochondria was once a separate organism. It was once a bacterium. Okay, mo na ilahang postulate. Okay, so take a look at this kaning gitawag nga quotation from Bill Gates. We know Bill Gates is a world-renowned um, computer scientist. Iya gi-revolutionize ang computer science when he developed this Microsoft. Di ba? And he's one of the richest person who ever lived. Okay, so according to him, DNA is like a computer program but far far more advanced than any software ever created. Okay, that's according to Bill Gates. Because I'm sure some of you have background sa kaning gitawag natong computer programming, and you know nga every single character nga inyong gisulat sa kanang code ninyo means a lot to the entire program. Okay, so say for example, inyong gi-place ang dot, okay, or comma, or unsa pa na mga apostrophe, depending on the what computer program you use. Okay, so i-remove na nimo, say for example you have 100 or 1000 lines and different characters na din ha. Kung i-remove nimo ang single character, okay, it can potentially paralyze the entire program, or it can make the program stop working. Okay, so that's what Bill Gates realized, that the DNA is also a code. Okay, the DNA is also a code. Nga every single movement is very specific. It's very precise. Nga if ever nga na ay mga, say for example, na ay mga kakulian or na ay mga kaning mga erroneous nga mga events, it can paralyze the DNA, or it can stop the DNA from doing its duties. Okay, so DNA information, take note ha, that DNA information is stored in chemical configuration. Okay, so code man siya. So when you say code man good, is naa siya gitawag natong information nga nature. Okay, so that information is stored in chemical configuration. So say for example, I'm pretty sure you are already familiar with this, the adenine to thymine and cytosine to guanine, and they are known as the base pairs. Okay, so mao na siya nga, 'di ba nga there is this consistent nga pairing of adenine to thymine and cytosine to guanine. So ATCG. And kaning base pair, so say for example sa usa ka DNA strand is naa siya ay pila ka thousands or even millions nga nagsunod-sunod ang ATCG, and then not really alternate, pero makita nimo is mura siya random, but it's not. It's very specific. Okay? Anyway, we are going to discuss that one further. So organized as chromosomes inside the cell. So when you say DNA, 'di ba, sa inyong genetics before nga gi-coil siya, okay, to become a visible structure, not really visible pero sa kanang gitawag natong compound light microscope, mahimo na siyang visible, 'di ba, kung imong because when you say DNA nga katong sa iyang loose nga configuration, di na nimo makita sa compound light microscope. But when they are coiled, they will form structures that are already visible under the microscope, known as the chromosomes. And it is very important nga they will be organized into chromosomes, especially when the cells are dividing. Diba? So dito na din na ma-connect din na nato sa mitosis and meiosis. Okay? So each species, take note ha, each species differ in the amount of information as well as the number of chromosomes. So take note that you cannot compare nga kung unsa ang amount of information sa humans and then i-compare nimo sa kuan. Yeah, you can actually compare, pero makita nimo didto nga there is this difference between the information from one species to the other. And take note, it's from one species to the other. Okay? So, ang number of chromosomes also, since na may difference sa number of information, so it can also result to the difference of the number of chromosomes. Say for example, human beings, di ba? We have 23 pairs of chromosomes ang human beings. When you go to ferns, na ay fern species nga tag 2,000 kabuok ang ilahang chromosomes. Nangiuban ari ka sa fruit fly. I think fruit fly only has 4 or 5 or 6 I forgot. Okay? But the thing is the number of information from one species to the other differ greatly. Okay? And if you are going to consider the difference between the number of chromosomes, so you can say that the number of information inside each species also differ. Okay, so there are different species with the same number of chromosomes. Okay. So di na to na tingala nga in the living world there are also organisms that have 23 pairs of chromosomes. Okay? So just like humans. So does it mean nga they are also humans? The answer is no, because the sequences of the base pairs differ even though that the number of chromosomes are the same, but the sequences of these base pairs are actually different. Okay, so I hope you get that one. Okay, so let's go to the chromosome. So this is now a little review of what you have discussed sa inyong gitawag na to nga genetics. So this is the DNA. Okay, we will start with this one. So this is the molecular configuration sa DNA: adenine to thymine, cytosine to guanine. You can see that one that it's paired. Okay, it's a double helix. Okay, and then it's very very thin. Okay, it's very very thin, pero it's very long. Okay, it's very thin but it's very long. Now if the cell is dividing, okay, it will be organized into coils. Okay, it will coil through the aid of this histone. Okay, it's actually a protein. Okay, and then coil further until it will become a chromosome. Okay, and this chromosome then can be located din sa imuhang nucleus. Okay, but remember that when cell division takes place, the nucleus disappears. Okay, the nucleus disappears. Okay, so that it will give way for the splitting of the cells. Okay, and I'm not going to discuss that one further. But the thing is, you notice here that this chromosome is actually made up of DNA, coil DNA. Okay, so kana siya. So what else? So the structure, okay, na ay different nga mga components of sa chromosome. Okay, you have the chromatids, kaning sister chromatids. This is actually during the time nga na S phase, di ba? So this is the kinetochore, okay, where the attachment of the sister chromatids, and the shorter arm is the P arm, and the longer arm is the Q arm. Okay? And the centromere is the centermost region of this chromosome. Okay, and they play a very crucial role, especially during mitotic cell division. Okay, so kana siya. And then the telomere here, and the telomeres are actually segments of this chromosome that protects the integrity of the entire chromosome. Okay, so take note nga when we are younger, okay, the telomeres are said to be thicker. But as we age, mao na nga it will become thinner, exposing a large part of the chromosomes. That is why it's no wonder why in the older age bracket, okay, dinha na manggawas ang mga quote-unquote genetic related na mga diseases. Okay? So, mao na siya nga ang telomere. Mao bitaw na nga nainangutanan nako nga kanang mas safe ba if tigulang ang lalaki nga kana bitaw kanang mohimo siyag anak. Okay, main ko nga okay raman compared sa babae, because we know nga if motongtong na gani og 35 hangtod or older ang babae, and then mamamdos pa siya, so taas og risk. Okay? At the same time, it's risky for both baby and the mother. Ingon siya nga. Then ingon dito kong friend nga, so safe ra ang lalaki. Inko nga, yeah, maybe there's a certain degree nga safe ang lalaki, but remember also nga the older the man gets, okay, the thinner is the telomere. So it's not just siguro ang risk para sa lalaki is minimal, pero the risk nga sa offspring nga mahimo is mo na murag crucial siya, na siya murag negative implication. Okay, so Mao na nga makahuna-una kag minyo og sayo. Okay. Pero I'm not actually kung ano yun, it's not no pressure. I'm just telling you the pros and cons. But anyway, I'm not a marriage counselor. Okay, so here there are different types of chromosomes, 'di ba? Na kay kanang gitawag natong metacentric. Okay, so naagid sa centermost ang centromere. Okay? And then you have the submetacentric. Okay? Medyo ang isa is taas, kini nga part, the upper part, the P arm and the Q arms are quite longer. So the metacentric, there is a relatively similar length sa P arm and sa Q arm. Then you have the acrocentric na ni gamay na gyud kay ang P arm compared sa Q arm. And you have the telocentric, okay? Pero it's not found in humans. Telocentric is I don't know kung gamay na gyud kaayo ang kan gitawag natong P arm, okay? Ang Q arm ang mas kaning gitawag nato nga prominent. Okay, so this is the human karyotype. So you notice here the human karyotypes. Remember that we have 23 pairs. Okay? 23 pairs. Okay, so mo na nga you have chromosome 1 hangtod nga naata sa 23. Okay? So when you say 23, the 23rd pair, this is now designated for kanang gitawag natong sex chromosome. Okay? So take note nga of course we have discussed this one in genetics. Take note that the XX will result to females and the XY is for the male. So in this human karyotype, we see that this is actually owned by a male. So mao bitan nagiingon nila nga naay kanabitang sa society nato karon kanang sa gender. So may say gender sa ilaha nga kuan, especially sa LGBT, gender is una nga choice, okay? But when you say sex, it's actually the biological nga kaning basis. So XX for the female and XY for the male. Okay, so kana siya. So this is now the cell cycle. This is just a little review of the cell cycle. Okay, so the animal cell, and we know that during the cell cycle there's a lot of genetic activity happening inside the cell. Okay, because remember, especially sa mitosis, the sister chromatids or not sister the sister cells must be identical. They should be clones. Okay? So kung pila ang nadawat sa isa ka cell after mitosis must be equal to what this other nga daughter cell nga iyahang na receive. Okay? And that includes the genetic material because we know nga if ever nga there is this inequality of the distribution of the genetic material from one cell to the other daughter cell, then that's not a good result. Okay, so we actually give credit to Gregor Mendel sa iyang discovery. Oops, sorry. Yan. We actually give credit to Gregor Mendel sa iyang study on genetics. Okay, so this is very common sa kaning gitawag natong chromosomal or the classical or the Mendelian genetics. Now, maingon ta nga dugay naman kay ng Mendelian genetics. Okay, pardon for that kay na fabrication na hitabo suloyo. Okay, when we say Mendelian genetics, even though that it was conducted centuries ago, okay, but the principle is relatively the same. Okay. So that is why, because of the work of Gregor Mendel, he was regarded as the father of genetics. And the sad thing is that his works were not recognized, okay, not until 35 years after his death. Okay, kana siya kay the story is that during the time that Mendel worked on his piece, that was also the time where Charles Darwin published his book The Origin of Species. Mao to nga na-overwhelm ang scientific community sa kung Darwin. They forget or they did not give that much attention to the work of Gregor Mendel. But that is why it's only after 35 years after his death that his work was rediscovered and were appreciated. Okay, so the molecular structure of DNA. Okay, so thanks to these guys, James Wilson, Francis Crick, Maurice Wilkins, and Rosalind Franklin, we have the accurate nga depiction of the molecular structure of the DNA. Now take note, before the results of the works of these guys here, okay, the DNA structure has already been known. Only that they cannot, there is actually a riddle, okay, na siya riddle, but they know nga na ay DNA, okay. And it was the work of Rosalind Franklin, ang kaning gitawag natong X-ray crystallography, that really affirmed that the DNA structure is a double helix structure, okay. And then these three guys here, they are actually chemists and physicists, okay. So mao na nga these three guys, Watson, Crick, and Wilkins, they were awarded the 1953 katong Nobel Prize. What about Rosalind Franklin? Rosalind Franklin died before the award. That is why there is no posthumous nga award given by the Nobel Prize. But she's of course vital to the discovery. This is actually her work nga nakapa-realize sa kaning tulo nga double helix, and at the same time run in an anti-parallel manner ang kaning gitawag natong DNA strand. Okay. Hope mahuman na dito. So these are actually, when we talk about the foundation or the structure of the DNA molecules, it's actually made up of three major components. You have the base, the nitrogenous bases. Say for example, in this case, it's adenine. Okay, this is the base. The second is the sugar. This is now, and the phosphate, the third. Kaning phosphate group and the sugar group are for the kaning gitawag tong strands. Okay, so again, these are the three: the nitrogenous bases, the sugar, and the phosphate group. So these are actually the major nga kaning, say for example, structure. Okay, the structure of a DNA molecule. Okay, so here, so take a look at this one here. So when we talk about molecular structure, the molecular structure, remember that adenine is always paired with thymine and cytosine is always paired with guanine. Okay, and this is for the DNA. And take a look at this one. Ang direction, okay, because they run in anti-parallel manner. So the direction runs from 5' to 3'. Okay, since anti-parallel man siya, so opposite. So ang pares sa 3' is 5'. Okay, and down, so 5' to 3', 5' to 3'. So that is what we call the anti-parallel nga strand. Okay, so take a look at adenine to thymine, cytosine to guanine. Okay, it's anti-parallel. Now, this is actually where This is actually where Watson and Crick got right. Okay, mao ni siya ang nakapakuan yun, nakapa-catapult nila sa kaning gitawag nato scientific fame. Okay, because ang sa una man god is before them they thought nga ang 3', ang pares sa 3' is 3' also. Okay, and then ang 5', ang pares niya is 5'. Pero by the time nga gitan-aw ni Watson and Crick ang katong X-ray crystallography ni Rosalind Franklin, they realize nga kung ato aning i 5' to 5' and 3' to 3', dili gyud mo match ang kaning gitawag nitong bonding, the hydrogen bond between the nitrogenous bases. Okay, so that's why niingon sila nga if ato aning i-run from 5' to 3' and then ang isa is opposite pod, okay, ay anti-parallel nato, is mo perfectly match niya. So it makes sense, everything makes sense. That is why it's very revolutionary. Ang ilahang trabaho sa correct structure of the DNA is very revolutionary because it paved the way for a lot of molecular biology nga activities, including the advances in biotechnology. Okay, so let's go now to the central dogma. Okay, so when you say DNA, we have discussed now the major nga nature of DNA. So there are different configurations of DNA, and genetic sequences is highly specific, di ba? ATCG, and then katong genetic sequence. Now there are segments that is responsible in the production of certain products sa cells. Okay, because you can think of cells as little factories inside your body. Okay, so sequences differ from one species to another. And take note that human genome has more than 3 billion base pairs, and more than 99% of that is similar to all humans. Okay? So kana siya. DNA in a cell. Okay, by the way, who are the people mangutan na ka, are there people nga 100% similar ang ilahang or 100% the same ang ilahang base pairs? The answer is yes. There are people who are having 100% the same ang ilahang genetic components, and they are the identical twins. Again, identical twins, not the fraternal twins. It's the identical twins. Okay, so DNA in a cell is 2 meters long in an actual size. So you can just imagine nga sa actual size kanang bitang microscopic niya nga kuan, kung imuha na siyang stretch, it's actually similar to a thread. Okay? Similar niya siya sa thread nga kung imuha na siyang stretch from one end to the other, kay linear man na ang DNA sa tao. Okay? It's actually 2 meters long. Okay? Sa isa lang na siya ka cell. Okay? Sa isa lang na siya ka cell. So, kung there are trillions of cells, di ba? If there are trillions of cells, and then they estimated nga kung ilay nimo ang kanibitong katong actual nga DNA length, okay, sa tanan nga cells imong body, okay, it can reach to the moon and it can still go back to Earth for 4 times. Okay? So yun ana niya siya kataas, and you can just imagine the amount of information nga naa sa imong cell. So ang kana man kung gitawag na tong kaning gitawag nato nga sequence, DNA sequence, muna siya book bitaw, muna siya libro sa ato ang life, sa ato ang body. Okay, say for example cookbook, you can just imagine cookbook nga say for example 300 pages cookbook. There are lots and lots of information about the how to cook, what are the ingredients in ana siya. So ang DNA nito is more than a library. Okay, more than the biggest library in the world, and you can just imagine the information on how to operate a certain cell sa kanang library. Okay. So when you say that central dogma, this is actually the common nga kanang gitawag nato nga depiction or the summary. So you have the DNA replication. This is the first. Okay. Next is the transcription from DNA to RNA, or from RNA ihang i-direct ang DNA. That's reverse transcription. Diba? Reverse transcriptase PCR, the RT-PCR. So, di na nagikuha. And then, the translation from RNA to protein. So, take a look at this direction. It's from DNA, gikuha ang information sa DNA, ang nagkuha na ang RNA. Segment lang, gamay lang. And then, ang RNA po, iya pong gitranslate into protein. Meaning to say, ang DNA ang product sa DNA, what is stored inside the DNA is actually protein. Okay? So DNA to RNA, RNA to protein. Now, can proteins make DNA? The answer is no. Okay? This is just a one directional nga process. Okay? Okay? So wala makakakita ani nga arrow nga reverse translation. No, you cannot do that. Okay? Only kini sila because maybe sa nature sa DNA and RNA, which are actually the nucleic acids. Protein is another biomolecule. So again, the end product of DNA is actually protein. Meaning to say, kanang mga genes kanang mga segments nga nasa tong DNA, they are actually coding for protein. Okay. So this is now the differences between DNA and RNA. Okay. Ang RNA is quite shorter compared to the DNA. RNA is single-stranded while DNA is double-stranded. Okay? And the nitrogenous base for DNA is adenine, thymine, cytosine, and guanine. And sa RNA is, instead of thymine, uracil. Okay? So, uracil, cytosine, guanine, and adenine. Okay? So, mauna ang gitawag nato nga differences, major differences between DNA and RNA. So, mangutan na ta, is there a double-stranded RNA? The answer is yes. Mangutan na ta, is there a single-stranded DNA? The answer is yes. Okay? Pero, in selected, very few lang na nga mga organisms ang nag-carry nga kana nga mga molecules. Sa case sa human beings is wala ta na. Okay? So, when you say DNA replication, this is step number 1. Okay? So, the amount of DNA doubles. Diba? So, DNA replication. Remember sa katong mitosis ninyo, during the S phase, the synthesis phase, okay, ni double ang amount of DNA. That is in preparation nga kanang gitawag nato nga mo split ang cells. Okay. So, it occurred during the S phase of the cell cycle, and it is semi-conservative replication. So when you say semi-conservative replication, remember, di ba, na ay double-stranded man ang imuhang DNA. So one strand is the original, and the new strand, the other strand, is a new. Okay? Kana bitang the original, ang isa is katong gi-synthesize nga strand. Okay? And it follows the 5' to 3' direction. Diba? Kato itong gi-mention ganiha nga the direction of replication is from 5' to 3', not the other way around. And it is participated by numerous other proteins known as enzymes, the group nga mga enzymes and other proteins. Because without the aid of proteins, it will take millions of years for a DNA replication to take place. And you don't have any millions of years. Okay? Because if muhulat pa ang DNA ana for millions of years, kuan na siya, madaot na ng DNA. So, sa enzymes, because of we have enzymes, DNA replication can be done in minutes. Okay? And the DNA replication is very specific, okay, and very accurate. Okay? Meaning to say, it should be 100% accurate, because kung naaga ni malian na, then it can lead to mutations, which is actually bad. Okay? So DNA replication is the doubling of the amount of DNA inside a cell. Okay? So this is actually the direction or a diagram of how DNA molecule replicates. Okay? So take note, it is from 5' to 3', diba? Ang katong direction sa katong atong gi-mention gani, it's from 5' to 3'. So mo start siya. So kaning blue, kaning dark blue, okay, this is the original strand, and it is semi-conservative, diba? When you say semi-conservative, is that the original strand is retained and then the other one is newly synthesized. Okay, so from 5', mao man ang direction 5' to 3', diba? Kay sa kuan man sila kanang gitawag natog anti-parallel. So ang pares sa 3' sa kaning sa dark blue is 5'. So since 5' na so dinhi mo start ang replication. Okay? So when you say replication, mo siya og zipper bitaw. Okay? We have here the helicase. Di ba kung zipper imong i-close, okay, mo sikit ang duha ka quote-unquote kanang strand. But kung imuhang i-open ang zipper, okay, kana bitaw na ay kanang imong gunitan i-close ug open, mao na ang function sa helicase, iyahan nang i-open. Once nga mo open siya, gikan pa man na siya dinhi. So once nga mo open siya, mosulod day ni ang kaning gitawag tong polymerase, and then pamoniton to niyang mga nitrogenous bases nga naa palibot sa DNA, iyahang ipares. So say for example, kining first nga kaning strand kini siya dinhi. Okay? So if this is adenine, ang ihang puniton dinhi sa imuhang kaning gitawag natong polymerase is thymine. Kung kini siya sa second nga base pair is guanine siya, ang ihang puniton dinhi is pares niya, which is cytosine, so on and so forth. Okay? Now, dinhi nga side, since anti-parallel man siya, so di ba mao ni ang 5', but remember that the direction of the replication is from 5' to 3', and since kining 5' is mao man ni siya ang gitawag nato nga kaning original nga strand. Okay? Dinhi mo sugod ang replication. Okay? So, inig-open ani niya, walay kaso sa kanang gitawag natong leading strand. Mao ni ang leading strand kay inig-open ana niya, panitan na dinhi ang gitawag nato nga naapa dinhi ang kaning helicase. So, inig-abli niya, so kanang replicate dayon. Igabli niya, butang, igabli niya. So, the more ni siya nga mo kaning helicase, the more siya nga mo travel down this kaning DNA. Okay? Musunod ra ni ang kaning gitawag tong leading strand. On the other hand, ang lagging strand, mag-agad pa na siya kung kanus siya mo abli. So, inig-abli niya, kung 5' ni siya, dinha, replicate. Mo forward na po siya, replicate. So that there is this what we call the Okazaki fragment. Unlike din nga tuloy-tuloy siya. Okay? But there is this mechanism. By the way, this is what we call the lagging strand. Okay? Nagsunod man ni, mas ulahi man ni siyang ma-fulfill compared sa leading strand kini. Okay? So, even though there is this kanang gitawag natong fragmented, take a look at this one here, the fragmented, the Okazaki fragment, even though ganay fragmented, but there is this what we call the DNA ligase. Okay? This is what we call the DNA ligase. So when you say the DNA ligase, mauna mosuroy na siya from this strand here, kini siya pangitaon to niyang mga mga unpaired, okay, unpaired nga mga base pairs, and then iyahang butangan o katong gitawag natong iyahang partner nga nitrogenous base. Okay, so dili na kaayo mabalaka. Now take note nga mubo ra ni siya kung kini siya, but in reality ang imuhang base pairs is by the millions, so taas kaayo ni siya. And you know what's amazing with this one is that it is in most cases 100% accurate. Okay, because the DNA kining replication, even though nga complex na siya, but naa siya proofreading mechanism. Okay, so i-proofread niya whether sakto ba ang gibutang sa imuhang ligase, sa DNA ligase or dili, because kung makita niya nga dili gani, then there is another mechanism nga iya hatong tangtangon ang mali nga pares, and then iyahang ilisan sa correct nga pair. Okay, so the overall replication is from 5' to 3'. Okay, so this is actually for linear. Okay, kung straight line lang ang imuhang gitawag nga DNA. What about the circular DNA, which can be found in kanang gitawag natong mga bacteria? Okay, so ang bacteria they have this circular DNA. So even though nga naay difference, but relatively the mechanism is the same, only that dinhi ang kanang gitawag natong difference. Okay, so kana bitawng duha ka buok, and then mo open na siya, mo split na siya, di ba, kay strand maghapon kanang double strand man gyud gihapon na siya. Okay, ang katong strand mo split gihapon to siya, take a look at this one, mo split gihapon na siya, take a look at this one here, nibulag siya, and then the same mechanism katong DNA primers, okay, and then mga helicase, the same ang mechanism niya hangtod nga magka-progress hangtod it will ultimately split or replicated ang kanang gitawag nato nga circular DNA. Okay, so it is semi-conservative. Again, semi-conservative, one strand is old or the original strand, and the new strand is the pair of the newly synthesized nga mga chromosome. So take a look at this one. Di ba? So ang kaning red kaning naa sa inner, there are the new strand, and the old strand is the dark blue. Okay. So, mo na siya ang overview na siya, though you can still learn, you can still read a lot of things about this replication. So, let's go to the mRNA. Okay? So, when you say mRNA transcription, again, this is actually getting, when you say mRNA transcription, this is actually getting the information from the DNA. Okay? Because even though nga ang DNA na siya ay protein nga code, okay? But oftentimes is that you don't need to express all the proteins at one time. So naray mga times nga ginagmay-gagmay lang ang expression, and the mRNA transcription will do the job. Okay, so mRNA, that's the messenger RNA. So you can just think of mRNA as the secretary. Okay, nga moingon ay mohang boss nga pangitaar gato sa chapter 1 maora toy ato ang i-discuss. So since ang sugo sa imong boss is only chapter 1, ang secretary is chapter 1 na po ito ihagikuha. Dili niya ipa-photocopy ang entire book, only the first chapter. Kaya maora may gi-require. So that's how I give the analogy for the mRNA. Okay, so there are types of RNA. You have the messenger RNA, the mRNA, the ribosomal RNA or the rRNA, and the tRNA or the transfer RNA. Even though that they are all RNAs, but they have specific jobs, and each of them have a very important and specific jobs to do. So, but in this case, let's focus more on the mRNA. So when you say the mRNA, the code from DNA is being read or transcribed by the mRNA. Okay? So only take note only specific segments of DNA undergo transcription. And we are referring to the specific genes. Mato kung gi-mention ganiha nga kung chapter 1 lang sa book ang ganahan sa imong boss nga DNA, ang imong secretary chapter 1 na pagyoy gamiton, that's the mRNA. Okay? So multiple transcriptions can take place at the same time. Multiple transcriptions but not all transcriptions. Okay? So remember nga ang imong DNA is a long thread. Siguro na ay gamay dinhi na po gamay din nag-transcribe, sa gamay na po din nag-transcribe, but not the entire DNA nga thread. Okay? And just like the replication, transcription must also be specific and accurate. So kung unsa ra gitoy message sa DNA, maora pod gito ang iyahang kuaon. Okay? So this is just a revisit sa genetic code. I don't worry, I'm not going to include this one. Okay? So if you are familiar with this genetic code, then that's much better. So this is the base, 'di ba? When we talk about transcription, later on na lang kay translation naman siya nga component. So ang kanang gitawag natong base is grouped into 3. Okay? We call that one as the codons. Okay? Sa imong DNA ba? So kung adto ka sa DNA na sequence ATTTT or CGGG, okay, it is grouped into 3. And each of those 3, okay, will correspond to specific amino acids. Okay? And there are 20 amino acids. I don't know if you have if you're done with biochemistry, but mano nga masumpay niya sa biochem, the nucleic acid. So each codon, okay, is mo correspond siya og specific nga kaning gitawag nato nga amino acid. So there are 20, and mao na nga ang isa ka protein will be characterized by the amino acid sequence. And asam naman nato makita dayon diin manigikan ang information sa amino acid sequence. It's actually coming from the DNA. Okay? So here, okay, so take a look this one. So this is where the DNA the mRNA transcription takes place. Kanilang nga segment mo open lang na siya. Okay? Sigin siya open ang tod nga mo lengthen pod ang kaning pagmanufacture niya. So just one strand, okay, ang iyahang i-copy. Okay? That is actually enough, and shorter nga segment lang. Okay? So kana siya, so naay similarity ang iyahang pag-transcribe sa replication, but similarity ha, I'm not telling nga it's the same similarity. There are actually deeper nga kaning gitawag nato mechanism, and I'm not going to discuss that one. Okay? And I'm not including that one sa akong quiz later on. Okay. So hinani okay, so this is the DNA. Okay, so this is as I have mentioned nga ang kaning imuhang sequence kaning ATGGGCC kaning siya, they are actually grouped into 3. Okay, they are known as the codons, and ang imuhang mRNA will transcribe the anticodon. Okay, again, ang imong mRNA will transcribe the anticodon. So unsa man ang anticodon or the antisense? When you say the anticodon, that is the pair of the DNA. Di ba sa usa ra man to ka strand ang mRNA transcription. So say for example, diri ta ni kuha sa 3' nga strand kining sa lower nga strand sa DNA. So the anticodon or the antisense of T is A. Di ba? Kay ATCG or AT and then CG. And then ang A, instead of thymine, 'di ba, ang sa RNA is uracil naman. So instead of A, that's U. Then C is paired with G, C paired with G, G is paired with C, G is paired with C, A is U, so on and so forth. Okay? So nakita niyo. So kung unsa gani ini siya dinhi, kung unsa ni siyang nasa ibabaw, you would notice that it is actually the same here. Okay? Except na lang sa kaning U, which is actually still it will still result to the same amino acid. Okay? So kung unsa ni dinhing naa sa ibabaw, di ba? Kaning sa 5 kani nga ibabaw nga strand. Okay? Di ba mo siya ang iyahang contemporary, iyahang pair. Pero kung tan-aw na to, kung unsa ni siyang naa sa ibabaw gyud is the same message here. Mo to kung gi-mention ganiha nga it is specific. Okay? It is just a secretary. And take note nga every three nga base nga kaning siya nga base is grouped into 3 kaning sila dinhi ang kaning imuhang mga sequence, mRNA sequence, and then ang kada group, okay, will correspond to an amino acid. So say for example ang AUG, that's 3, that will correspond to methionine. Okay, so nga how do we know nga methionine here? Let's go back to the genetic code here. That's AUG. Say for example AUG here, this is methionine. Okay? So, kag ang methionine, it can also be kuan if AUU, AUC, and AUA, that is also no, I mean, AUA and AUG is methionine. So, kung ka og AAU, that aspergine. Okay? Kana siya. So, hangtod adto nga if it is already translated, manon man yung translation, we are now entering translation, that is now known as the peptide sequence. Okay? So, no longer sa katong gitawag nato nga nucleic acid sequence, but now the peptide sequence. And take note that peptides are now the foundation of proteins. So, we now go here sa protein translation. Okay? So, when you say protein translation, the message from mRNA taken from DNA is being read and translated into corresponding protein. Basically, the message of the mRNA is the same as the gene from the DNA that is being copied. Hence, what is written in the DNA is eventually turned into protein. So, that's translation. Okay? I-translate nimo ang unsay message. Okay. So, here, so this is now the transcription, and then giread siya sa ribosome. Okay, take note, ang ribosome, di ba, sa cell bio ninyo is ang ribosome, the one that synthesizes protein. So dinhin na siya. So siya ang tig-copy. Okay, ang ribosome. Siya ang tig-copy kung unsa ni siya nga codon, and then siya ang tigbutang sa katong gitawag natong mga amino acid hangtod nga nakay polypeptide chain here. Okay, hangtod nga mahuman niya og basa ang tibuok nga mRNA, which is actually shorter compared to the DNA. Then you have now the primary structure of the kaning gitawag nato nga protein. So this is now the structure of the amino acids. Take note that there are 20 amino acids. Okay, 10 of those are essential and 10 are non-essential. So what's the difference between essential and non-essential? When you say essential amino acids, those are the amino acids that our bodies cannot synthesize. Okay, dili manufacture sa itong body. That's why we have to eat this. We have to look for sources for these amino acids mo na nga nasa food, and particularly sa kaning mga beans, di ba, cauliflower kan siya. So di na nato makuha ang atong amino acids. Okay, so take note nga when you say transcription, it happens inside the nucleus, and once the mRNA mo na nga dapat shorter and single strand ang mRNA aron makalusot siya sa sa katong gitawag natong nuclear pores. Okay, so ang transcription happens inside the nucleus. So with the kanang itong DNA replication, ang mRNA after ana is mugawas siya sa imuhang nucleus, and naa siya sa cytoplasm, dito siya mahitabo ang translation. Okay, so this is now the imuhang tRNA, which is actually working closely with the ribosomes. Okay, ribosome is actually a protein based na siya. Okay, so ribosome, protein synthesize siya sa protein. Okay, so this is now the polypeptide. So the longer you go, okay, the farther this tRNA, okay, the longer is the polypeptide chain. Okay, so this is how it is done. So take a look at this one. This is your ribosome. Actually, there are two subunits here. Okay, this is your tRNA. Your tRNA is bearing kanisya na siya mga particular nga mga gitawag nato nga amino acid. So say for example, ang UUU, it corresponds to the amino acid lysine. Okay? So how do we know? Let's check the genetic code. UUU. Here. Oh. Di mo asa man to? Is that correct? Oh no, this is the kuan. Mauni siya ang lysine, ang AAA day. Okay, kay anticodon man. But anyway, I'm not going to discuss that one. Kay I'm not really including this sa inyong kuan genetic code. Okay? So, asara to. lysine. Ako sa tong pangitaon. Wala man. But anyway, I'm not going to kuan. So mao na siya nga as it moves forward, tan-awan nimo nasumpay ang duha. Okay? Nasumpay ang duha hangtod nga kini pod another tRNA Iyahan nang i-attach din ha, and then musumpay na po na siya din hangtod it will become longer. So here, now take note nga mo to akong gi-mention ganiha nga you have this primary protein structure. Okay, katong kan. Now, take note that proteins will undergo series of foldings, molecular foldings. So, hangtod na because kini siya nga primary structure, this cannot be utilized by the cell. Sa atong body, di niya ma-utilize. I-fold-fold pa na siya. So, na kay secondary structure, the tertiary structure, and the quaternary structure. Mauna ni ang gamiton sa ato ang body. Magsige na siya fold. And you can just imagine nga ang naay mga protein nga tag 2,000 kabuok amino acids. And kung naay isa ana nga sayop, okay, useless or kanang gitawag natong destructive ang gitawag nato nga kaning protein nga na form. Okay? So, take a look at this one. So, unsa may application aning gitawag natong central dogma sa biotech. Okay? So, insulin from animals sa unang panahon is from pigs. Ang kanang mga isa, special sa mga diabetic kanang gina-inject. So, ilang squeeze tong pancreas and then man nga nakay tambal. Pero karon unsa na may karon? This is actually before biotech. So pero karon, take a look at this one. Okay? Nagkuha ka og isa ka segment or gene sa humans, gi-attach nimo sa circular DNA sa plasmid, okay? Extrachromosomal DNA sa plasmid, okay? Na nakay recombinant plasmid. Okay? So meaning to say, gi-insert ni siya balik sa bacteria, ang bacteria na dayon ang ni-produce og human insulin. So nakita nimo ang application. So mo akong gi-emphasize sa inyo nga it is very important nga when you study biotech is you also have to understand the molecular basis of kanang gitawag tong genetics because dinha gyud nag-base ang majority sa biotechnological nga kaning activities. Oh, mana. Okay. So, I think that's the end of our discussion. And if ever nga you do have questions and confusions and clarifications, you can actually write it sa comment section or you can message me directly or through sa atong Google Classroom. Okay, so I think that's all for today and I hope you learned something. Enjoy every moment of your life. Enjoy it with your family and your friends, your loved ones. Okay, and learning shouldn't be a burden. Okay, so God bless us all and see you next meeting. Bye-bye.